Welcome students. Let's start our topic cell membrane. In this topic we will discuss fluid mosaic model of cell membrane. Let us students start to discuss now fluid mosaic model. Fluid mosaic model of cell membrane is most accepted model which is given by two biologists Singer and Nicolson in 1972. So it is very important to note down that Singer and Nicolson had given the most accepted model of cell membrane. According to this model, students, the membrane is not uniform disposition of lipids and proteins, but is instead a mixture of the two. So it is very important point that was missing in first two models. Here it is clearly said that cell membrane has lipid and protein in a mixture, not present at a proper positions. Next point, the lipid component of the membrane is mainly consist of either phosphoglycerides or phospholipids, and carbohydrate present in cell membrane are either branched or unbranched oligosaccharides. Next students, in the cell membrane, lipids are arranged in such a way that their polar heads are directed outward whereas non-polar tail are directed inward. Student, it is very important to note down that the polar head are hydrophilic, that is water loving, whereas the non-polar tail are hydrophobic. So what we can say that arrangement is in such a manner that non-polar tail can be protected from the aqueous environment. The structure of the phospholipids or lipid is similar to the structure of soap that we have discussed in our 10th class. Here the diagram that I have drawn, the head, that is rounded one, that is directed toward the outer side, whereas tail of the two respective lipids are facing each other. So this internal area is basically the non-polar one that is occupied by the tails of two phospholipid layers. Next student, membrane proteins are also present in case of this cell membrane, and the membrane protein can be classified as integral or intrinsic proteins and peripheral or extrinsic protein. So what does it means? That the protein which are present in cell membrane that are of two types, external one called extrinsic or peripheral, and internal one which are present internally in the cell membrane that are called integral or intrinsic proteins. Peripheral proteins lie on the surface of the membrane, that is on the peripheral surface, whereas integral proteins are partially or totally buried in the membrane. So what we are getting from this slide, that the lipid which are arranged, that are arranged in a such a way that their tails are facing each other whereas heads are directed toward the outer side. And second student, the proteins which are having in the cell membrane that are of two types, one is extrinsic or peripheral proteins and second is integral or intrinsic proteins. Let us try to understand fluid mosaic model with the help of diagram. So as per the diagram students, there are two phospholipid layer. This is one and this is second one. And what is special with them, that their head are facing outward and head are tailor polar whereas tail are non-polar, and being polar, head are always hydrophilic whereas tails are always hydrophobic. So what does it means? That the internal area where these non-polar tails are present, that is hydrophobic. Beside this student, there are membrane proteins. Some are peripheral proteins which are present on the outer surface, and some are internal proteins which are either partially buried into the cell membrane or completely buried into the cell membrane. Those membrane proteins which are internal and completely buried in the cell membrane, they are called tunnel proteins or transmembrane proteins. Please note down that those integral proteins which are buried completely into the cell membrane, that are called either tunnel proteins or transmembrane proteins. Beside this, students, some other structures are there. Carbohydrates are there in the form of oligosaccharides, and if some sugar molecules are attached with proteins or lipid molecule, then they are called respectively glycoproteins or glycolipids. So very easy to understand from the diagram that according to fluid mosaic model, phospholipid bilayer is there in which heads are directed outward whereas tails are facing each other. And beside this, membrane proteins are there. Some are present externally, some are present internally. The proteins which are present externally, they are called peripheral or extrinsic proteins, and those which are present internally, they are called internal proteins. Now we know that internal area is hydrophobic, so sometimes these transmembrane proteins or tunnel protein help the water to enter into the cell because they provide channels so that the water molecules can pass through these channels. So very important function transmembrane proteins are playing, that they provide channels for the entry of the water into the cell membrane. Next, student, cell membrane is quasi fluid, that is not solid. So it enables lateral movement of proteins within the overall lipid bilayer, and the ability to move within the membrane is measured as its fluidity. Whatever the ability that proteins have to move, flip-flop movements they are showing, basically they are moving here and there in the lipid bilayer. So this is measured as its fluidity. Next, student, the fluid nature of membrane is also important from the point of view of functions like growth, formation of intercellular junctions, secretions, endocytosis, and cell division. So you can say that quasi-fluid nature is helping proteins to move here and there, which is giving it cell membrane the fluidy nature, which is responsible for cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. Students, protein molecules occur at places both inside and outside of the lipid bilayer, which appears like protein icebergs in a sea of lipids. So this is a very important line, that why this term is there, because protein molecules occur at places both outside and inner side of the lipid bilayer, and thus it appears like protein icebergs in a sea of lipids. So students, if we talk about the functions, cell membrane or plasma membrane perform various functions. But here students, we are mainly concerned with the most important function that cell membrane plays, that is transport of the molecules across it. So there are various methods that cell membrane is opting to allow transport of the molecules. So let us discuss first method, that is passive transport. In case of passive transport, students, molecules move from higher concentration to lower concentration, that is across the concentration gradient or along the concentration gradient. And if these molecules are solutes, then the process that is used under passive transport, that is simple diffusion. And if the molecules are of water, then that is also a diffusion, but because here the water is crossing the semi-permeable membrane, then that is not called diffusion. That diffusion is specially called osmosis. So what is student passive transport? Here the molecules move from higher to lower concentration along the concentration gradients. Neutral solutes are moving through simple diffusion, whereas if water is there, then it is crossing semi-permeable membrane, then this diffusion is called osmosis. Students, as we know that polar molecules especially the water that cannot cross the non-polar lipid bilayer. So for polar molecules like water, if they have to cross, sometimes some protein that are present in the membrane that help to pass that molecules into the cell membrane. And if proteins are helping polar molecules to transport across the membrane, then this diffusion is called facilitated diffusion. So facilitated diffusion is also one of the method of which is covered under this passive transport. So what is facilitated diffusion? When some proteins are helping polar molecules to move through the non-polar lipid bilayer. After passive transport, student, now second method is active transport. Active transport is utilized for the transportation of ions or molecules when they are moving against the concentration gradient. That is, when they are moving from lower concentration to higher concentration. And students, very important point to be noted down, that such a transport is energy dependent process in which ATP is utilized, and it is called active transport. For example, sodium and potassium pump that playing a very important role in the body, that utilize this method of active transport. So it is just opposite to passive transport. Here things are moving from low concentration to higher concentration, and it is a energy dependent process. Here large amount of energy is utilized in the form of ATP. Lastly, student, third method is bulk transport. This process involves lesser amount of energy as compared to active transport, and it involves especially endocytosis. Endocytosis include two main terms, that is pinocytosis. Pinocytosis means cell drinking, and phagocytosis. Phagocytosis means cell eating, and exocytosis. Exocytosis is also commonly called cell vomiting. So what is bulk transport? Here in this process, unlike active transport, lesser amount of energy is utilized, and it involves endocytosis and exocytosis. So here we have discussed some methods of transport of the molecules across the cell membrane. We have discussed three important methods, that is passive transport, active transport, and bulk transport. So in whole discussion, in today's discussion, student, we have talked about fluid mosaic model, in which we have seen that fluid mosaic model has phospholipid bilayer, which is also containing proteins which are either present on the surface externally or either present in between the lipid bilayers. Besides, proteins also have some carbohydrates, especially oligosaccharides, and some glycolipids and proteins. In NCRT diagram, we can also see that beside this all molecules, some cholesterol particles are also shown. So some cholesterol particles are also associated with cell membrane. And lastly, we have discussed about some method of transport across cell membrane, that is passive transport, active transport, and bulk transport. So student, with this, I'm going to close our today's topic. In the next class, we will meet with the new topic. Till then, take care. Thanks. Have a nice day.